Hi everyone, welcome to our webinar today. My name is Rebecca Northeast and I'm a product manager here at Proteintech. I'll just wait a minute and wait for some people to join live and then we'll get started. I hope you're having a really great Monday and I wonder how many of you will be joining us for the rest of the immunoassay week. So tomorrow, I'm leading the IHC and IF workshop, and then on Wednesday we have the immunoprecipitation workshop, and then on Thursday we're doing another one for Western blot. So it's a really busy week here for us at the events team at Proteintech, and we're really excited to see more of you. If you would like to join these meetings, we'll send you the link further on for the other meetings if you're interested, if you missed them before. So do stay tuned. I think it's gonna be a really exciting week. And with that, I'll get started on today's talk. So today we're going to be talking about antibodies, for sort of how antibodies are made commercially, why antibodies are used in life science research, and then also talk about the importance of antibody validation, how that is affecting life scientists all over the world, and why it's really important that you, the end user, are aware of how antibodies are validated. So really briefly, I'm going to start with a quick company introduction. So Proteintech is a leading life science reagent company, and we have products which comprise across 3 core product lines. So through our Proteintech brand, we sell our antibodies and immunoassays, and we're predominantly known for our antibodies, and we have these against 13,000 human targets. We have over 400 ELISA kits, IHC easy kits, and then through our humankind brand, we sell our recombinant cytokines and growth factors, and these are HEC293 expressed, which is a human cell line, meaning that it has native folding and post-translational modifications and also higher bioactivity. And then through our Chromatech brand, we sell our nanobody-based reagents, and as the name suggests, these are much smaller than your typical antibody, which makes them excellent for immunoprecipitation of tagged proteins, as well as things like super-resolution microscopy. As a company, we measure our success by measuring our customers' scientific success, and we're incredibly proud to have been cited in over 200,000 times worldwide, and we are now one of the top 10 antibody companies by citations, and probably the fastest growing company in product citations as well. So what am I going to talk to you about today? So first, we're going to start with an introduction to popular immunoassays. So anything pretty much that most lab scientists do at the lab that involves an antibody. Then talking about commercial antibody production, how antibodies are made and how that makes them differ in terms of their type, and actually why that's important to the end user as well. Then really briefly going to touch on controls for immunoassays, why they're important. Then talk about the reproducibility crisis and the role that antibodies have had in this crisis. And then talk about antibody validation best practice and how Proteintech is validating our antibodies. So let's start with the antibody itself. So antibodies are some of the most common reagents used in life science research, and they are produced by the body as part of the adaptive immune response to an unrecognized antigen. They are a hugely powerful tool in research, diagnostics, therapeutics. When I tell my non-scientist friends that I work for an antibody company, they all go, oh yeah, COVID, antibodies. So antibodies have kind of had a bit of a renaissance. Everyone really knows what they are. They're not necessarily aware of how they're used in research. But for example, we have all these mAb treatments. Anything that has any drug name that has mAb at the end, that is a monoclonal antibody target. So they're pretty much ubiquitous in all of science, our antibodies. So now I'm going to talk through some of the most common immunoassays, why they're used, the differences between them. So probably the one that everyone knows and is most sort of cognizant of is of the Western blot. So Western blot is used as a method to detect proteins in a highly sensitive manner. Western blots also allow you to detect any post-translational modifications, any mutations, relative expression. It is a qualitative and a semi-quantitative method. So it's mostly relative, mostly qualitative. You're looking at it, say, relative to a housekeeping protein. It enables you to detect proteins in a protein lysate preparation. So you do have to prepare the lysate in order to then run the Western blot. It can allow multiplexing, but typically not always used in a multiplexing manner. And it is a slower, relatively complex protocol compared to others. But one of the many joys of Western blot is that it's quite cheap to run and it is accessible to many, many labs worldwide. IHC stands for immunohistochemistry, and this is whereby we're looking at the spatial localization of target proteins, typically using chromogens. So this can be highly sensitive. It's also always qualitative. It's quite rare that you can do quantitative IHC. It requires specific tissue preparation, which can be relatively complex and also relatively tricky to do, but there are lots of labs and cores out there that allow you to do this. It can be high throughput when automated, and IHC, for example, is used for looking at prognosis in cancer patients and all sorts of other things in the clinic, as well as in research. It is relatively slow and complex protocol, but like with Western blot, it is typically done and available to most labs. Next, we're talking about immunofluorescence. So immunofluorescence is where we look at the localization of a target protein using fluorophores. And because the use of fluorophores, it means that we can have really easy multiplexings. This is where you get your really beautiful images, lots of different colors. This is typically immunofluorescence. And again, it is slow and relatively complex, but also available to most labs, possibly less so than IHC because you do require fluorescence microscope or a confocal microscope. But again, most labs worldwide are able to perform immunofluorescence. Next, we talk to the ELISA. So the ELISA is a very different beast in comparison to the other three. However, what it does do is that it's very quick and it allows you to have highly sensitive detection of an analyte, most typically in a crude sample preparation. So here we can run things like saliva, plasma, serum, CSF, all sorts of samples that don't have to go through a protein lysate preparation, and you can get a very quick quantitative answer about the presence of this protein in your sample. It's very quick, simple, high throughput. Most people can run this. You essentially just need a kit and a plate reader, and it's used in the clinic, it's used in research. It's a really hugely popular application. And then lastly, just talking about flow cytometry. Flow cytometry has had a huge boom over the past 10 years. It's incredible because it enables you to analyze protein expression on an individual cell basis. So you essentially need to create a single cell suspension, and then utilizing lasers and a series of relatively clever mirrors and PMTs, so these fluorescence detectors, you can then detect the different levels of proteins on your cells, and then that enables you to assess the different cells in a heterologous mixture, typically done with things like immunology, where you want to look at different subtypes of immunology cells in your patient sample or in your research sample. So I'm not going to go through this, but this is just sort of a table explaining and showing the differences of immunoassays. So that was just a whistle-stop tour of all the immunoassays. Well, not all of them. There are so many out there, especially now with all these really high throughput, multiplex biomarker tools available to you. So antibodies are an incredibly powerful tool. But now we're going to talk about commercial antibody production and why this matters and why end users should be aware of this. So in terms of polyclonal antibodies are by far the most popular and widely used type of antibody today. This is partly because the way that they're made is that you use a long protein immunogen, and then you collect all the antibodies that are recognizing this immunogen. So that means that your polyclonal antibody is a mixture of IgG. So it's a mixture of antibodies that recognize a variety of epitopes across your immunogen. So because we have many antibodies recognizing many targets across your protein of interest, that means polyclonal antibodies have a higher total affinity to your antigen. They are much more sensitive, so you can detect lower abundant targets. They also cross-react with many species. So if you're, for example, a researcher looking in zebrafish or looking in xenopus larvae, things like that, there aren't many antibodies that are directed against these proteins. So instead, you're using human antibody proteins, things like that. So if you have a polyclonal antibody, you're far more likely to recognize a rarer species than you are with, say, a monoclonal. These are also excellent for capture. So if you're trying to capture a protein out of a solution, things like ELISA or immunoprecipitation. One of the slight negatives of polyclonal antibodies is they do have batch-to-batch variability due to the production in different animals, and I'll talk about that on the next slide. And also they have an increased chance of cross-reactivity with the protein that you're not expecting. And that's because, as I mentioned, they are recognizing across the whole protein immunogen. So you are more likely to have potentially some cross-reactivity with a similarly structured protein, but not the one you're actually trying to detect. So how are polyclonal antibodies made? So first off, we start by immunizing an animal. So that's typically a rabbit or a chicken. And what happens then is you then undergo a booster program. And when that animal is ready, you then collect its serum. And then when you have that serum, you pretty much have all the total antibodies that that animal has produced. So what we then need to do is purify those antibodies against the immunogen, so just against the target protein that you're trying to create a commercial product to. So we do that by having our bead, which they mobilize antigen on, and then we purify these antibodies. And so you get a sort of antigen-specific mixture of your polyclonal antibodies. And this is why we say that our antibodies are affinity purified, because instead of just providing researchers with this mixture, we've actually provided them with a highly concentrated mixture of antibodies that are specific for the target protein. And this is sort of where the lot-to-lot variability with polyclonals come in, because as you can imagine, there is unfortunately, no such thing as sort of an immortal animal, an immortal rabbit. So you will have to start immunizing separate animals to start creating different lots in order to have sort of commercial viability and keep producing the antibody. So this is possibly where you get lot-to-lot variability. It might be that you have a slightly different result between lot-to-lots. Companies like Proteintech, when we do make different lots of polyclonals, we undergo loads and loads of testing to ensure that the performance is as equivocal as the lot before. But you can also write to certain companies like Proteintech and ask them to reserve certain lots for you if you find one that works much better. So that is something to be aware of as well. Companies will do polyclonal lot reservation if you ask them. And in terms of polyclonal antibodies, just a note about the types of immunogens that you can use. So the immunogen is the protein you inject into the animal to generate the response to then collect the antibodies. So in terms of Proteintech, we always try and use recombinant full-length or long-length proteins. And this is because historically, many antibodies used to cut corners and use short peptides as their immunogens. And this would generate okay antibodies for linear epitopes, so things like Western blot. However, these were suboptimal for other applications like IHC or flow cytometry, where you're trying to recognize the protein in its native form. So at Proteintech, we always use full-length recombinant antibodies, and this therefore enables antibodies that can be used in more applications and have much higher flexibility and success. So next, we're going to talk about our monoclonal antibodies. So as the name suggests, instead of poly, we've got mono, and that means that they are only recognizing a single antigen epitope on that protein. And so a single epitope, if you're interested, is 5 to 8 amino acids long. And monoclonal antibodies are really good because they enable low background, because you're less likely to have cross-reactivity. They're good for high abundant targets. As you can imagine, they have slightly less affinity and less sensitivity. So they're really good if your protein or antigen of choice is in high abundance. And they also have minimal lot-to-lot variability. So they're good for long-term projects if you don't want to get any issues between, perhaps if you go back and try again, but that antibody lot has changed, you want to be in monoclonals ensure minimal lot-to-lot variability, so you've got better long-term results. One of the downsides, as you can imagine, is decreased total affinity and also limited species reactivity. Because they're only 5 to 8 amino acids long, there is a chance that with a slightly rarer species, those 5 to 8 amino acids aren't in the homologous portion of the corresponding protein in a different animal, different species. So how are monoclonal antibodies made? So monoclonal antibodies are typically made by injecting a small animal such as a mouse. And then once that immunization process has undergone, we then remove the spleen and isolate the B cells and plasma cells from the mouse. And then these isolated cells undergo hybridoma creation, whereby you fuse them with an immortalized tumor cell, and this then creates the hybridoma. We then screen every hybridoma by ELISA to check really briefly for its possible signal strength for reaction with the target immunogen. Then we take our chosen clones and they undergo what's called clonal expansion. Then we run more tests, and then from one of these clones, we then choose that as the commercial monoclonal antibody. And what's really important to note here is that we're now not using any animals. We've got this sort of immortalized cell line, so we can keep producing as much monoclonal antibody as we need. We're not relying on nature anymore to generate the antibodies lot to lot. We instead have this in vitro cell line. One thing to note as well is that even though this is obviously much better than the polyclonal method, however, sometimes you can get genetic cell drift. And companies like Proteintech, we have lots of ways to ensure that this isn't happening, and we're always really rigorously testing our lots in between batches. But that is just something to be cognizant of about monoclonal antibodies, is that you can get genetic line drift. So next we're talking about recombinant antibodies. So recombinant antibodies have been touted as the future of antibody manufacturing. And this is where you go one step further from monoclonal antibody production. And you then take the antibody gene and you clone the heavy and light chain into an expression host. So we have we now know the genetic makeup of the antibody itself. So that means that we no longer have any lot-to-lot variability because we can produce the same antibody again and again in a recombinant manner. And also, because we know the sequence of the antibody binding domain, we can then also try genetic engineering to try and improve its specificity and affinity. And this is typically done for therapeutic antibodies. You can do this in an in silico manner, or you can do this otherwise. And so if you are really interested in recombinant antibody production, it's something that's really interesting, and it's definitely how all the kind of commercial therapeutics are done for antibodies. And at Proteintech, we are rapidly expanding our recombinant antibody range. We're aware that this is essentially the future of antibodies, and we're really, really excited to be in this space and to be working towards expanding our recombinant offering. So how does Proteintech make our recombinant antibodies? So what we do is we first immunize a rabbit, and then we isolate its B cells. And then once we've selected the B cells that are going to go up into becoming the commercial product, we then clone the heavy and light chain into the expression vector, co-transfect this into a suitable mammalian expression system, purify the antibodies, and then validate it further before it's ready to release. So this is how you get your recombinant monoclonal antibody mixture. So really briefly, just when we're talking about recombinant production, Proteintech's nanobodies, which are a alpaca-derived nanobody, which is essentially the binding domain of the heavy chain only antibody from an alpaca. And these are then used for things like nanotraps, so immunoprecipitation, or nanoboosters, so for immunofluorescence. And these are always recombinantly produced. So again, you get pretty much the exact same performance lot to lot with the Proteintech nanobody portfolio. So I won't go through this again, but this is just a summary slide dictating the different types of antibodies available to you. Essentially, if you're not sure, polyclonals are really good for things like low abundant targets, rare species. If you have a cross-link fixative like IHC, monoclonals are good for longer term projects, high abundant targets. If you're having high background with a polyclonal, it can be really good if you switch to a monoclonal. You will then get lower background. And the same can be said for recombinants. They're really good for long-term projects and also excellent reproducibility. So just so you guys are educated and aware of different types of commercial antibodies available to you. So now really briefly, I'm going to talk about controls for immunoassays. It's a really important note for immunoassays that we include the correct controls, because what we want to make sure as scientists is that the data we are reporting out from our antibodies is real biological signal and not actually background noise or off-target binding. So the ways that we can do this is doing things like a secondary only control. So what this does is this will reveal anything that might be signaled due to off-target secondary antibody binding, and ensuring that all the signal we're seeing is actually only due to primary antibody binding. The endogenous control is really important to reveal any autofluorescence or sample artifacts that might appear as signal. And then next we have our antigen controls. So our antigen controls would include a positive control. And this is where the sample is well known to express the target. So it proves that your protocol is working, the antibody is working as expected. And then your negative control is where you have a sample where there is no protein expression or very little protein expression expected. And that will help reveal any false positives or non-specific signal from the antibody. If you're short on time, and especially for things like immunofluorescence, IHC, secondary only and endogenous only controls are really important when you're first trialing a new antibody or when you're running a different sample, just to ensure that you're aware of the expected signal to see, and you can troubleshoot from there if you start seeing any unexpected results from these controls. So next I'm going to talk about the reproducibility crisis and the role that antibodies have been having on this reproducibility crisis. So I'm sure I don't have to tell a group of intelligent scientists like yourself that life science is undergoing a reproducibility issue. And it's really well known and been published sort of since like the early 2010s. This is just a survey showing that a huge percentage of people, I think it's like 80% of scientists that were surveyed, think that there is a life science crisis, a reproducibility crisis. And one thing that is considered a big culprit, a big cause of this, is poorly validated antibodies. And this is, for example, where end users are assuming that vendors have validated their antibody. They see something with high citation and they assume that means it's had good validation, but this isn't necessarily the case. There is also a general lack of awareness in the scientific community. The problem with poorly validated antibodies and poorly validated results anyway, reagents anyway. So what can happen is you've got well-meaning researchers finding relatively interesting or unexpected results with an antibody, and then they spend thousands or millions of dollars following that up, trying to find out where it's going. And all that's happened is that it's essentially a result that was off the back of an antibody that was not recognizing the protein that the researchers thought it was. So as laid out by Begley and Ellis in Nature, these are some of the reproducibility principles that life science should be run by. So things like studies should be blinded and randomized. All results should be shown, so you're not just commonly emitting inconvenient data. Experiments should be repeated, and also positive and negative controls should be included. So we were talking about that in terms of antibody, in terms of our immunoassay controls. And the fifth one is that reagents should be validated. Often that's not done and not omitted. Nowadays as well, some journals are asking more and more for evidence of reagent validation, like antibody validation. So do be aware of that and think about why it's really important to your results. Next one is sort of the cost of bad antibodies. So as I said, you can have people following spurious results. It's leading to essentially failed clinical trials. There are many published examples of this. Global spending on antibodies is huge. It's over $2 billion per year. Yet some papers are suggesting that only 50% of these antibodies have been properly validated before use. And there's one really good example where a researcher was looking at 13 ER-beta antibodies. So ER-beta is a popular treatment prognostic marker for breast cancer. It's really widely used globally and for oncology research and oncology diagnostics. And they found that only 12 out of 13 of these antibodies were cross-reacting with other proteins. So there was only one that was found to be specific for ER-beta, and this one was actually the least publicized and the least used. So it's just something to be aware of, that just because something is well used and well publicized doesn't necessarily mean that it's the best antibody or the best validated antibody out there. So one possible issue that could be occurring in this antibody crisis is that because of how antibodies are made and antibody production, many companies actually do not make their own antibodies in-house. So what they do is they purchase from other suppliers, but these people are not disclosing who they're purchasing from. This is called OEM, by the way, in the industry. If you ever hear someone talk about OEM, it means original equipment manufacturer, and that's when you're buying in another product from a different manufacturer to sell it under your label. So as you can see here, what could be happening is that the end user or the customer is buying the same antibody, but it's just labeled differently. So they're buying it from multiple companies. So they're essentially wasting money, wasting time. They're getting the same poor results. And one other point to note is that sometimes original validation data cannot be provided on request. Say you come up with a complaint to your antibody provider, they aren't able to show you the original validation data, the original production data, because they weren't the ones that made it, and the manufacturer won't disclose it to them. So this is where Proteintech came in, and it was one of our founding aims, was to make every antibody ourselves and to validate it ourselves and to ensure that it's working in customers' hands. So we are essentially cutting out the middleman. You can never buy another antibody under another Proteintech antibody labeled differently, and we will never buy anyone else's. So it's not like you're buying, you can buy a different one from us. We can provide you with the original validation data upon request. We're incredibly transparent with customer reviews online and with our citations. And also because it is all complete in-house manufacture, we have complete control over this. So we're able to recall any bad lots, we're able to produce more in line with global popularity. So it means that your supply chain is much more stable as well for the end user. So how did the scientific community come to reply to this and have with all these issues in the poor antibodies and poor antibody manufacture? So in 2016, there was this creation of an international working consortium for antibody validation. And they developed 5 conceptual ideals for how antibodies should be validated. And there was also big calls for antibody registry databases for transparency. And I'm going to spend the rest of the talk talking a bit about this. But just briefly, these are the 5 sort of ideals that first came up in 2016. So orthogonal strategies are where you're trying to detect a target and then compare it with an antibody-independent method. So that might be things like you've got your antibody with your protein with your antibody, and then you're comparing it with mass spectrometry or transcriptomics. And then for antibody-independent strategies, this is where you're comparing antibody activity with that of another antibody that is binding at a separate epitope. So, for example, 2 monoclonal antibodies that bind at separate epitopes. You can then check that you're getting the same response in both. Genetic strategies are very commonly used. So this is where you're checking that the antibody is working in, say, a knockdown or a knockout genetically. Recombinant expression is where you're looking at things like a tag protein. So you're seeing, you can look at, say, a GFP tag, and you're seeing the same band or the same response when you detect an antibody against GFP, as well as an antibody against the target native protein. And then capture mass spec. This is where the antibody is immobilized on a bead and used for target capture. And then any bead-bound proteins are subject to mass spec analysis. So that was a whistle-stop tour. I have actually included all the papers at the end of this if you're interested in reading it itself. I'm now going to talk about best practice and how Proteintech is able to validate our antibodies in line with these ideals and in sort of our own enhanced validation, because the ones that I showed previously, they're not always necessarily the best, and us and others have shown that there are other ways to validate antibodies that show it's specific, and how we're ensuring that the antibody is ready to use when it comes to you, our customers. So we've got 8 sort of ideals here, and I'm going to go through each of them. First off, starting with application-specific validation. So application-specific validation is actually really, really important, and not every manufacturer does this, but what we're ensuring is that by testing every antibody for relevant applications for that target, we're ensuring that it's working in every hand. We know that not every antibody that, say, works for Western blot is the best antibody for flow cytometry or the best antibody for IHC. So what we're doing when we're validating our antibodies is we're showing the user where their antibody is good for and how we do that. So one of the things that we look for when doing antibody-specific, application-specific validation is we first want to understand the target. So there are loads of amazing databases out there like UniProt, and that will help. It provides all the sort of literature available to you about that protein. It will tell you where it's located in the cell. It will note all its biological relevance, and it will also tell you of any post-translation modifications to be aware of. Next, you then need to identify the cells and tissues you want to validate it in. So you want to confirm expression of the target in that. So you can do that with literature reviews or through other methods. You can then identify any positive or negative controls for the validation. And it also, you need to ensure that your antibody is then going to work in native samples. It's all very well generating an antibody against an overexpressed protein, but that's something that's actually very easy to do. Antibodies against highly expressed proteins are really quite easy to make because of its affinity. So we really want to make sure that all our antibodies are working in native samples, not just overexpressed or recombinant protein. Next, we're then going to choose the appropriate applications for these antibodies. Like I said, not every protein that is suitable for flow cytometry is suitable for IHC, for example. And then we're going to ensure that there's a transparency of results. So as I said before, we're going to display all our validation data online. We can provide you with any extra validation data on request, and we're always ensuring that reviews and citations are accessible. So really briefly, how do we validate our antibodies for Western blot? So we first look at its predicted molecular weight, and then we can account for any predicted post-translation modifications. So we know, for example, how much a phosphate group and a sugar group glycosylation will add to the weight of an antibody, and also the presence of any isoforms. So this is a really good example here. This is the ZAP protein, which has 2 well-predicted isoforms, which come out really clearly in our validation data. So you can see them with these arrows. We also talk about the different protocol conditions. So there are some quite tricky proteins to analyze by Western blot, especially membrane proteins. So we'll provide you with any of the reducing condition changes that are required to properly visualize that by Western blot. For example, boiling or no boiling for the membrane proteins. Next one is IHC. So IHC, we validate our antibodies for ensuring that it's the predicted tissue and cellular location, and also correct co-localization where that's suitable. So this is a really beautiful example here of insulin and glucagon cells. So as we know, insulin and glucagon aren't produced by the same cells, so there's no overlap here, but they are produced in the same location in the pancreas. So you've got this sort of next to co-localization, showing that these antibodies are specific and acting in the way that we expect. And one thing as well that's really important for IHC is antigen retrieval. Not everyone is aware of this, but antigen retrieval is so important, especially when looking at cross-linked fixatives. So we will then also detail when we're doing our validation, which antigen retrieval is best for this protein in that tissue. Next one is immunofluorescence. So again, immunofluorescence you can validate by just assessing the correct cellular location. So in this really beautiful image, we're staining for TDP43, which is green, which is, we know, a nuclear protein. In red, we've got mitochondria, which we can see is outside of the nuclear envelope. And then lamin B1, which is in cyan. You can see the nuclear envelope, which is really beautifully outlining the nucleus here. So we're really well aware of all these antibodies producing the correct patterning and showing that they are actually valid for immunofluorescence in this way. We also look at the different fixatives, because different fixatives, especially for cells and immunofluorescence, can have a massive impact on how that antibody performs. So, for example, here these are most likely PFA fixed, but if you're looking at an extracellular epitope, then things like ice-cold methanol is a much better fixative. And then just lastly, talking about immunoprecipitation. So when we're looking at application-specific for IP, we're confirming that the antibody can bind to native antigen in the lysates. We recommend the amount of protein, the amount of antibody to get a sufficient pull down. And we also recommend an elution buffer that suits that protein best. And then lastly, talking about flow. So flow cytometry, first you want to research the target. Is stimulation required in order to visualize the target, get a specific staining, a significant staining bump by doing the stimulation? And also, really importantly, you want to know where the immunogen is localized. So you want to know if you're looking at an extracellular target or an intracellular target, because that will then determine whether or not you need to do any fixation and permeabilization. This fix and perm as well also depends on the size of the protein, that if it's intracellular. And then in terms of determining its performance, we can do what's called the separation stain index to determine its working concentration. So now we've talked about application specific, we're going to really briefly talk about genetic strategies. So genetic strategies for antibody validation are really, really popular by many antibody companies. And this is because it provides a really clear answer as to whether or not your antibody is specific for that target protein. So firstly, we'll talk about knockdown. So knockdown is where we're looking at things like RNA interference. So that might be si or sh RNA. And what this does is this clears the target protein mRNA and therefore reduces expression. So what happens is you get massively reduced signal in your Western blot or IF, for example. So it's best for proteins with high turnover. And as I said, signal should be significantly decreased, as you can see in this example here. You can also nicely see it in this HADHA antibody example, where the cytoplasmic signal has almost totally gone in the siRNA knockdown. Next is genetic strategies. So genetic strategies are things where we look at CRISPR-KO. So these are either in cell lines or animal knockout models, so we can look at animal tissue. So really nicely here, we can see in the peroxidoxin 1 antibody in knockout, you can see that there's no signal here. And the same for our TDP43 recombinant antibody. In the knockout cell lines, we're seeing absolutely no signal. These data were provided to us by Icarus, and Icarus are an independent antibody validation company. And I'll talk a bit about them later, but I am next going to show a few examples of how our antibodies have been independently validated by this, this charity, Icarus, over here. Other sort of knockout examples you can see, I think I mentioned animal tissue before. This is a really nice example of SIRT2, which is present in the cortex of this mouse brain, but in the SIRT2 KO mouse, it's no longer there. And the same for our TDP43 antibody. This is a mix of knockout and wild type cells. In the knockout cells outlined by red here, there is absolutely no signal, again indicating the specificity of the antibody. One other really neat way that we can identify antibody specificity, if you don't have access to knockdown or knockout cell lines, is by using endogenous controls. So we can consider endogenous controls as sort of the simplest form of positive and negative control. So we can use things like PAXDB in the Human Protein Atlas, to find cell lines or tissue with really high expression. So for this IL-22 receptor antibody, you can see that the mouse small intestine has a high amount presence, but we know that it's not present in the spleen. So this again acts as an internal endogenous control, and that's just a really nice example here. Next, as well as sort of doing all the genetic strategies, we can look at how the protein is expected to behave. So what can happen is we can then do some experimental manipulation and check whether or not the signal does change in accordance to the manipulating that we've done. So, for example, in this ACE2 monoclonal, we know ACE2 is highly glycosylated. So if we perform deglycosylation with PNGase F treatment, we can see that there is a really big shift downwards in the size of our ACE2 band, indicating that yes, it has been removed for its deglycosylation. And same for phosphorylation. So when we're looking at phosphorylation antibodies, we can induce phosphorylation with things like calculin, I can never say that, with this molecule, this treatment here. And what that does is that induces phosphorylation. And you can see in the untreated cell line, there's absolutely no signal. So we're not recognizing any of the phosphoprotein. But when we're looking at the treated cell line, we can nicely see the presence of the phosphate band here. Next, we're talking about biological response, and this is a really neat way to, again, test how an antibody's specification. So this is HIF1 alpha. So HIF1 stands for hypoxic inducible factor. So as the name suggests, this protein is sensitive to hypoxia. And what it does is it translocates the nucleus in hypoxic conditions. So these are untreated cells versus hypoxic treatment cells. And you can say really clearly that this protein has moved towards the nucleus. And the same here, this protein is now really highly expressed following hypoxic treatment. Again, with biological response, there's so many ways that we can look at this. So I did neuroscience myself as my PhD, so I did a lot of CFOS treatment. So I'm aware that CFOS is a neuronal activation marker, and it becomes really highly expressed during stress. So this is the mouse cortex. This is data provided by a customer at the University of Sheffield. And you can see under basal conditions, there's really quite low expression of CFOS, but following restraint stress, you can see a lot more strong signal of this CFOS activation marker. And again, with caspase 8, under staurosporine treatment, you can see that there's a much higher expression of the cleaved caspase 8 versus the 8. So again, these are all kind of markers that the antibody is performing, the protein is performing as we expect, and therefore we can reliably say that the antibody is specific for that target. Next, we're going to talk about multiple antibody comparisons. This is, again, one of the ones that was on the pillars. So here we've got two different antibodies that are recognizing different epitopes, but you can see really nicely in the same sample. So NUN is a really common neuronal marker, and you can see really nicely in this mouse cerebellum tissue, it's producing almost identical staining. And then another way that you can do that as well is with things like immunoprecipitation. So you can do the pull down with one antibody and then detect it in the immunoblot with another. And if you're seeing sort of the same size, then that is a good way to indicate that your antibody is specific. Next, we can briefly talk about antigen cellular locations. This is, again, a really nice, easy way for us to determine if an antibody is specific. So you've got things like NF-kappa B and P65, which is predominantly cytoplasmic under normal conditions. So when we're running the nuclear versus the cytoplasmic fraction, you can see there's much stronger signal in the cytoplasmic fraction versus the nuclear fraction. And the same for ND2, which is a component of the mitochondrial complex 1 subunit. And what this does is when you run the whole cell fraction, you've got much weaker signal compared to the same amount of protein loaded for the mitochondrial fraction. And sort of one of the last things I want to talk about is, so I've talked a lot about antibody validation, explained why it's really important. And I've given this talk quite a few times now, and I've often had people ask me, well, what can I do? How do I validate antibodies myself? So one thing that we can talk about is, obviously, you should have your due diligence when purchasing a new antibody. You want to check, is that antibody validated for your specific application? Are there any customer reviews? How well cited is it? And do you trust these citations? And also, do you trust your antibody manufacturer? Do you know where it's come from? Once you've purchased your antibody and you've made that decision, what can then you also do in your own hands? So first off, you should look at, if you can, doing a positive and negative antigen control. So if you look at the antibody validation data from the company, they might have, say, tested it in this certain animal tissue or cell line, because we'll have done all the research to show that it is actually expressed in this cell line. And you can then confirm it with the literature. So then when you run your positive and negative controls, you'll be able to determine whether the antibody is specific in your hands, and also the protocol is working in your hands as well. One thing as well I really recommend, and from my time in tech support, I think I know a lot of people aren't doing this, but when they get a new antibody, they're not necessarily checking which antibody dilution is best for them. So what we really recommend doing is serial antibody dilutions for each of your different sample types and for each of the different applications. So here we really recommend, sorry, I'm worried my laptop might die. We really recommend that you do, for example, 8 lower or, and then go up serially from that. And then also do keep notes of and record all batch numbers for your reagents. So that's, again, really important. Next step, I want to talk about working with Icarus. So Icarus is an antibody characterization through open science. They're a company, a charity actually based in McGill in Canada. And what they're doing is they're trying to characterize every antibody available against the human protein. And it's, like I said, it's open science. So they're publishing their results. And Proteintech is working with them. Us and other leading antibody companies, we're providing them our reagents, and they're therefore characterizing them and then publishing the results online. So you can see some of their really beautiful results here. But if you are interested in sort of how people are doing antibody characterization, then Icarus is definitely one of those. Other antibody validation initiatives, so you can look at RID identifiers, so antibodypedia.com, antibodyregistry.com. What these do is they give unique identifiers for every commercial antibody out there, so you can then quote these when you publish. Journal guidelines nowadays are now asking more and more for antibody validation. That's either if you've done it yourself or if the company has done it, so you could provide them with Proteintech's antibody validation, say knockdown data. And also there's a big stress now to educate users. So for you guys to attend talks like this, to understand how antibodies are made and understand the differences between them and why antibody validation is really important as well. So these are some of the references I made. We'll send these out to you later on. I just sort of wanted to mention this is sort of a Proteintech slide. So we are a scientist for scientists. We have the largest in-house portfolio of antibodies against the human proteome. We're manufacturing them all, which I said why that's important. You can't buy a Proteintech antibody labeled differently. And we've been really fortunate to win various awards for our work as well from SciTab. And just really lastly as well, I wanted to talk about our antibody labeling kit. So this enables you to multiplex antibodies from the same species. And it's also compatible with any primary antibody and ready in as little as 10 minutes. If you're interested, do check out our website. And also, everyone that attended today is eligible for a free sample. So you can try one of our antibodies, one of our nanotraps, or one of our human clone proteins. If you're interested, you can either scan this QR code now, or we will send you the link. And with that, I think Amy is going to be on and ask any questions, if anyone has any.
Hi, Rebecca. So yeah, that was a really great presentation. Thanks everyone for joining. We actually just had one question come through from Anne. She says, can you give me a practical way of selecting the right secondary antibody for immunofluorescence? So which type of antibodies are most suitable, monoclonal, polyclonal, or recombinant?
Yeah, that's a really good question. Secondary antibodies typically are polyclonal, and that's because you want to try and recognize as many epitopes on your primary antibody as possible to try and get that signal amplification and get best signal. It's actually a really poignant question to ask us, because we have just released our recombinant polyclonal secondary antibodies. So typically recombinant secondary antibodies are made in things like the goat. Because of its larger size, you can get a really high output of secondary antibodies. But we've now gone down the route of creating recombinant secondary antibodies. So they are a multi-mix of different clones reacting against different epitopes of your primary antibody, but because they're recombinant, you're going to get really good lot-to-lot variability.
Great.
Good question.
We've got a couple more questions coming in as well. So one from Maria. How are antibodies for phosphorylated antigens generated?
That's a very good question, and that's pretty much a whole PhD thesis. But we do have some resources online. But what we do is they are essentially, it's quite a short peptide, and it's a phosphorylated peptide that we then use as the immunogen. If you are interested, then do feel free to email me and I can send you our phosphorylation resources, because unfortunately I don't quite have the time to go into it. But essentially what we use is a phosphorylated peptide as the immunogen.
Well, and we've got just one more. So are endogenous controls only necessary for immunofluorescence?
Sorry, what did you say?
Sorry, we've got one more question. So are endogenous controls only necessary for immunofluorescence?
No, you can run endogenous controls for pretty much anything. Typically Western blot, immunofluorescence. Imagine for things like flow cytometry, it might be a little bit harder, but Western blot is definitely a really nice one. For example, I did neuroscience, so I would know that the hippocampus has certain abundant proteins in it that aren't present in the cortex. So you could just run different lysates for that and just check that you've got low versus high signal where you expect.
Perfect. So I think that's everybody's questions that have come through. I think your presentation was so clear that we haven't been inundated too much. But yeah, thank you very much.
Okay, fantastic. And yeah, please do check out the rest of the talks that we have this week. And any questions, do feel free to either email myself or directly or the events inbox, and we will get back to you. Thank you very much.